So I can just do it a little bit later. Okay, it's not big. So I'm gonna ask you.
I know, we could let her know, because Susanna can't cry this morning. So then, we're gonna find one at 9. Yeah, not. 7, 6, 5.
Good morning, everybody. Kind of a dreary morning, but I guess we've been spoiled. A lot of nice days. Oh, goodness. Anything that I have to say this morning? Don't think so. Do you guys have any questions? Missing 3 people today. Well, I know where one is. So. Okay. Shall we get cracking? All right, yes, the obligatory picture of my grandchildren and my children and first meeting of the brothers whose birthdays are 4 days apart, you know? Yeah. Yeah. Cody's sitting there eating something. I don't know what it is.
Chicken nugget.
But anyway. All righty. Enough of my gushing. So, just a few things to clean up here on topic 6 before we jump into the cardiovascular system. Last couple of drugs I want to talk about, hallucinogens. A couple of pretty popular, well, one very popular one now, one that was very popular when I was your age. So cannabis, marijuana, and lysergic acid diethylamide, which is LSD, which was the psychedelic drug of choice when I was in high school. So there you go. And both of these things kind of have some impact on the brain amines. And I say particularly norepinephrine and serotonin here, but as you'll see in a moment, cannabis actually does impact dopamine as well. So, you know, just like the other drugs that we've looked at, these things kind of mess with these key neurotransmitters that are impacting brain function. So just a few notes about LSD. LSD is a really interesting drug in that it can really get into the brain quickly, and that is a result of it being lipid soluble. But what's even more interesting is it doesn't get out of the brain very fast. It just kind of hangs around. And humans are really sensitive to this stuff, susceptible to doses as small as 20 micrograms. Okay, so it's really small. Back when I was a kid, you know, there were a lot of stories about people getting high on LSD because they would lace the backside of stamps. Back in those days, we actually licked stamps to put them on envelopes. Most of you probably have never even done that. Yeah, we used to have to lick stamps to put them on envelopes. It's not one of these peel and stick things. And so people get LSD highs that way. So as far as mechanisms, so I'm going to jump down to this one. We really don't know exactly how LSD works. There are a lot of hypotheses out there. So some think that it stimulates serotonin production and inhibits serotonin uptake. The inhibiting of the uptake of serotonin is a bit controversial, so that may or may not be the case. A newer hypothesis is that LSD will upregulate glutamate release in cerebral cortex, which is where the major excitation is occurring that causes some of the symptomology. But the main outcome of LSD is that it creates such a heightened state of awareness in the brain that really teeny, what I term here, insignificant things just turn into these monumental, impactful sorts of things. Somebody comes up and says hi, and it turns into World War III. I mean, it's really quite a powerful drug, and it's actually made a little bit of a comeback. People, its use has seen a little bit of an uptick in recent years, but it is pretty nasty stuff. Okay, so for those of you who have printed out slides, I added a few because historically I've not talked a lot about marijuana, but since it's something that is fast tracking in this country to becoming totally legalized, I figured we ought to say something about it this morning. So I've added a couple of slides.
Are these slides on Canvas?
Not at the moment. Okay. Because I added them this morning.
Okay, cool.
So I added them to my presentation this morning, but I will update Canvas.
Okay, perfect. Thank you.
Okay, and actually, when you downloaded from Canvas, did you download a file or did I give you a link? It
takes you to Google Slides, and then
so all I need to do is just copy the version that I don't work on these things in Google, because I find that files get corrupted really easy when I'm working on them in Google Drive. So I work on them locally and then I re-upload them. And so as soon as I re-upload them to Google Drive, they'll be available.
Okay, perfect. Thank you.
And that's something that takes me no time to do at all. Okay. So anyway, so cannabis or marijuana is a psychoactive drug, surprise, that is produced from the marijuana or cannabis plant. The active ingredient is tetrahydrocannabinol, THC, as we like to call it. And while this is the main psychoactive component of marijuana, I found it interesting that there are 483 known compounds that come from this plant. Yeah, and it's not the only, and THC is not the only one of these compounds that is psychoactive. So I thought that's kind of interesting. It has a whole bunch of physical effect. We are actually still learning about all the things that cannabis does, because to be honest, the amount of research that had been done on it was relatively small until all these states started legalizing it, and now the research is like busting out of the seams because people are really wanting to try to understand what the long-term effects of the use of this stuff is going to be. But in terms of some of the symptomology for recreational use, euphoria, altered states of mind, and sense of time, you don't concentrate so well, your short-term memory gets impaired, and it influences your balance and fine motor control. So very much like alcoholic intoxication, it can be relaxing, and it does increase your appetite. If you are engaging in heavy use, the symptomology is a little more severe. So this can be high anxiety, hallucinations, panic, paranoia, and various sorts of psychoses. So if you use a lot of it, the stuff can have some pretty nasty effects. Anyway, it is also highly lipid soluble, so getting into the brain is not an issue for THC. And what's really interesting is that even after you use it just once, levels of THC can be detected in the body for weeks. So it's it's not one of these things that, you know, if you're trying to avoid a drug test, you know, you don't want to use it and then expect it to be gone a couple of days later, because it won't be. So. Okay, so as far as the mechanism of how it works, it's the primary thing that it does is it is an agonist for a couple of if I can get my lips to say this correctly, cannabinoid receptors, CB1 and CB2. Both of these are G coupled or G protein coupled receptors. The CB1 is the big one in the brain, and the most important one that THC interacts with. There are some CB1 receptors in peripheral tissues, but most of them are in the brain. The second receptor, CB2, is primarily in peripheral tissues, but I found this interesting. It's also expressed in neuroglial cells, which means you can find it in the brain as well. Anyway, primarily what it does is that it alters, you know, it basically creates all of its symptomology as an agonist against the CB1 receptors. And what it does is it inhibits secondary messengers, primarily adenylate cyclase. And, you know, the inhibition is going to be stronger the more of it you take. So, you know, that's kind of how it works. So, any questions? Threw all that together this morning for you. Yeah.
You said that THC lasts like several weeks in the body. Does it still have an effect afterwards? Like,
well, if it's there, I can see no reason why it wouldn't. But when it says it can be detected, doesn't mean that there's a lot of it. I'm sure that over time, you know, it degrades to, or it decreases to levels that probably are not going to have a huge symptomatic effect on you. But the key thing here is it can be detected for a long time. And so, you know, if you are a marijuana user and, you know, back before days where it was legal and you were getting tested for this, it was really hard to hide the fact that you'd used it, because it just hangs around for so long.
yeah, that makes sense. Thank you.
Okay. All right, one more slide. I thought I would throw something. I put this in a year or two ago, and I figured I might say something quick about NSAIDs, which are things like aspirin and ibuprofen, since, you know, they're drugs that we all use periodically. These are non-steroidal anti-inflammatory drugs. That's kind of what NSAID means. So I'm going to focus on ibuprofen here. It works in kind of an interesting way. It actually blocks the production of prostaglandins, and specifically prostaglandins that rear their ugly heads when you get injured. And these prostaglandins are going to drive things like inflammation and pain and swelling, you know, those sorts of things. And so it blocks their production, and that's basically how it works. And so, like, I just got my fifth COVID shot. I had the bivalent shot last Friday. And when I was feeling really achy on Saturday, I took Tylenol instead, because the last thing that you want to do when you want your immune system to actually beef itself up is to take an anti-inflammatory, which is what ibuprofen is. So there you go. So anyway, its specific mode of action is it inhibits enzyme activity, specifically cyclooxygenase, or COX. And this prevents the conversion of arachidonic acid to prostaglandin H2. And so that's the big thing that it does. And prostaglandin H2 is a big prostaglandin that's involved in these sorts of things in this bullet point here. The interesting thing about these NSAIDs is to really work, they've got to get inside your central nervous system. And they seem to be able to do that. And, you know, they're still studying the mechanisms by which this works. But the bottom line is NSAIDs, and ibuprofen in particular, is transported across the blood-brain barrier by proteins. There are transport proteins that will that will conjugate with the drug, and will shuttle them across the blood-brain barrier. So there's actually a transport mechanism that is built to get these in there, and that transport mechanism must have other functions, because I'm sure that, you know, it didn't develop in our systems just so that we could take ibuprofen. So there you go. So anyway, so that's a little bit on NSAIDs. Questions? Yeah.
Sorry, you mentioned that the other drugs like enter the brain quickly, leave slowly. How does ibuprofen work in that sense?
You know, I honestly don't know how quickly it leaves, but it, you know, it lasts for several hours.
Yeah.
So I'm guessing it doesn't get out that easily.
Okay. Thank you.
And then they'll say it'll be metabolized after a while. So anything else? All right. Topic 7, circulatory system. Let's deep dive into the cardiovascular system. So a lot of stuff we're going to talk about initially, if we're going to be probably review for most of you, because we got to kind of set a structural landscape for some of the other things that that we are going to talk about. First off, why the heck do we need a circulatory system? You know, when you look at a single-celled organism, you know, like this amoeba right here, no need for a circulatory system, because they can do all the transport they need by, you know, diffusion or other sorts of assisted transport mechanisms. But if you're a multicellular organism, such as these cute critters right here, you're really too big to mobilize things that need to get around the body by simple diffusion. It would simply be too slow. And so when you reach a certain level of complexity, you really need to have a circulatory system to help you get things from one place to another in the body. You know, circulatory systems come in 2 broad flavors in organisms. The simplest are circulatory systems that we see in a wide variety of invertebrate organisms. These are called open circulatory systems. And so the tissues inside of invertebrates are often bathed very directly with their blood, which we tend to refer to as hemolymph. And the cavity in which all of their organs exist, we call it a hemocoel. And the way that the system works, they have the hemocoel, where all of their organs are, is going to contain up to about 40% of their total blood volume. But the way these open circulatory systems work is you've got all this blood just sloshing around in the hemocoel, and then when the heart contracts, it pushes blood through the vessels. And then when it relaxes, it's got valves on the outside that will literally suck blood up from that hemocoel back into the heart to be moved via circulation. So circulation will be carrying it through the lungs or whatever, you know, to oxygenate it. And then when it gets to the end of the blood vessels, which are open, which is why they call it an open circulatory system, that blood will just be dumped directly onto the internal tissues, where the blood will do what blood does. It will exchange gases and pick up waste products and so forth. The big downside of an open circulatory system is they operate at low pressure. Because the vessels are open, you know, they can't generate really high pressure. So these types of circulatory systems are only going to be found in organisms that tend to have lower metabolic rates. Although there are some organisms, and I think specifically a certain insects like bees, that can actually, for periods of time, generate pretty high metabolic rates, yet they will still have an open circulatory system. Okay, everything else, including us humans, have what we call the closed circulatory system. And that means all the blood in our circulatory system is enclosed within vessels. We don't get any direct bathing of any tissues or anything like that. And being enclosed within vessels means now we can generate higher pressures, which makes it much easier to move the blood throughout circulation. All vertebrates have a closed circulatory system. In humans, just to give you an illustration of the extent to which circulation permeates our bodies, have in the neighborhood of 60,000 miles of blood vessels. Wrap your head around that. 60,000 miles. And that includes all the capillaries and everything else. But when you think about it, every cell in your body, which is getting nourished by circulation, has to be close enough to a capillary for that to happen. Isn't that pretty impressive? Okay. The circulatory system, although we call it a system, is so integrated with every other system in the body. In fact, there are many who argues that teaching physiology from a systems perspective is really not the best approach, simply because systems just tend to be so integrated with one another. And circulation is probably the extreme example of this, because no system can function without an integration with circulation. None, not a single exception to that. All right, so circulation is the monorail of the body. It transports everything that the cell needs and removes everything that the cell doesn't want. So red blood cells are the cellular or solid component of blood that moves around in our blood vessels. It's going to transport oxygen to the tissues and carry away carbon dioxide, as well as be involved in things like pH management. We'll learn more about that later. It's going to transport nutrients that are absorbed during digestion. Blood will carry these through the liver first, because this is where any detoxification has to occur. You kind of protects the rest of you from interacting with things that are not really good for you, and then to the tissues after it goes to the liver. It removes metabolic wastes, excess water. So if you're drinking a whole lot of water and you overhydrate yourself, then you got to get rid of that, because your body wants to stay in water balance. Then circulation is going to take care of that as well. It's going to get rid of any extra ions you have that you don't need. Pretty much anything that you don't want in your system, circulation will carry it somewhere to get rid of it. A lot of that will get filtered in the kidneys and be excreted as urine. Carries hormones. By definition, any biological messenger that is secreted by a gland and delivered to target tissues by circulation is a hormone. This is why when we talk about hormones a little bit later in the class, we will see that hormones can be basically any kind of molecule. There's not one type of molecule that makes up the hormones. They can be lipids, they can be proteins, anything that is going to deliver a biological message via circulation. Circulation participates in immunity and protection. So it protects you from injury, it protects you from invasion by foreign bodies, and it also eliminates toxins that may get introduced. So, you have white blood cells that are going to be involved in immunity. You've got clotting mechanisms that will repair punctures in your blood vessels. All of these are associated with circulation. All right, let's talk about the central piece of circulation, the heart. The heart is a double pump. It's a double pump that provides circulation to two separate circulatory loops in your system. The heart is divided in half by a septum, and the right and left sides of the heart each serve to pump blood through each of these different circulatory loops. There are 4 chambers in the heart. The 2 upper chambers are small, thin-walled. We call them the atria. They basically are designed to receive blood that is returning to the heart. And then the big players in circulation are the larger lower chambers, which we call the ventricles. We have a right and left ventricle. These are very thick and muscular, and that's just because when they contract and move blood through circulation, they have to have sufficient power to overcome the resistance that is offered by the blood vessels when you're trying to push fluid through. There are 4 major vessels that serve the heart. The vena cava, the aorta, the pulmonary artery, and the pulmonary vein. And we'll talk about what each one of those do. So the aorta is the big boy here. It's the largest vessel in your body. It is also the vessel that experiences the fastest flow rate of blood. We'll talk about that in a little bit, not today, but a little bit later as well. Okay. The aorta carries blood away from the heart. In fact, probably have that definition here. Arteries, the aorta is our largest artery, are vessels that carry blood away from the heart by definition. And the aorta carries blood away from the heart out of the left ventricle. And the left ventricle, coming out of the left ventricle through the aorta, the blood is being sent to what we call the systemic loop. Basically, it's all the tissues in your body except the lungs. I already said that. The vena cava is a vein. And the purpose of the vena cava is it is the final vein that will return blood to the heart from systemic circulation. So the first circulatory loop that we're talking about is the systemic circulation or systemic loop. And so the vena cava is going to return blood to the heart into the right atria. So if we look at the heart right here, we have the aorta, blood coming out. It goes into systemic circulation here. It does everything that blood needs to do out to all your tissues. It returns via the vena cava into the right atria right here. Veins, by definition, are vessels that return blood to the heart. So as the shortly after the vena cava, shortly before it actually joins with the heart, you have 2 major branches of the vena cava that will come together, the superior and inferior vena cava, that basically supply the upper and lower half of the body, and then they will join and empty into the right atrium. All right. Then from the right atrium, the blood is going to drop into the right ventricle. And when the heart contracts, blood from the right ventricle will leave the heart again into the pulmonary artery, which will deliver blood, again, carrying blood away from the heart. It will deliver blood to the lungs. We refer to this as pulmonary circulation or the pulmonary loop. So this is the second circulatory loop. The pulmonary vein then will return blood from the lungs to the left atria of the heart, where the process will start all over again. So in the lungs, the blood's going to get oxygenated. It's going to get rid of CO2 and be prepared to again make a pass through the systemic loop. Any questions to this point? This should probably be reviewed for all of you, I would imagine. Okay, so the main thing that I want to point out here actually is down here. This is a cartoon diagram of a cross section through the ventricles of the heart, and I just want to illustrate how the left and right ventricles are not symmetrical. They are different. The left ventricle has much thicker muscular walls than the right ventricle does here. Anybody want to offer a reason why? Go ahead, man.
Because that's to create a lot higher blood pressure to get blood from much longer distance.
For what, which one?
The left ventricle.
Yeah. So the left ventricle has much more vascular resistance that it has to overcome than the right ventricle does. The right ventricle just has to perfuse the lungs, and so it doesn't need to be as powerful as the left ventricle does. So that's why they're asymmetrical. Here's a chart that kind of illustrates in a human how blood is distributed amongst a variety of broad tissue categories. So we see the brain has 13%. It's not the largest, but when you consider the amount of blood that it gets relative to its size, that's actually a stunning amount of blood, and just emphasizes just how important circulation is to the brain. The heart itself generally only has about 4%, skeletal muscles 20%, skin 9%, kidneys 20%. Kidneys, you know, every time it passes through systemic circulation, the kidney's going to see everything. So your abdominal organs 24%, and everything else 10%. In total, the average human being has roughly 5 liters of blood total. And that's just that is an average value. Males tend to have larger blood volumes than females do. I'll give you some specific figures for that a little bit later. Oh, one other thing I'll say about this, these numbers are resting values. Okay, if you are engaged in exercise, these values can change substantially. Okay, so regulation of blood flow through the heart, that is accomplished by a series of valves. You have valves that exist between the atria and the ventricles. We call these atrioventricular valves, and there are two of them. Between the right atrium and the right ventricle, we have the tricuspid valve. And between the left atrium and the left ventricle, we have the bicuspid valves, and they're named because of the number of flaps that the valves are made from. And I'll actually show you an overhead picture, an overhead diagram of these in a moment, but you can see them. You can see them right down here. These are really neatly designed because you have these chordae tendineae down here that are attached to the interventricular wall, so that when the ventricle contracts, these flaps will be pushed upwards, and those chordae tendineae will keep the flaps from kind of inverting up into the atrium, and it makes a really nice seal, keeping the blood from going where it isn't supposed to go. There are also valves at the entrances to the major arteries. We call these semilunar valves, and they're called semilunar valves because if you look at them in profile, you know, they sort of look like crescent moons. And the way that they work is, you know, the blood pushes them up, and then they come down, they fall against each other like so, and it forms again a nice seal. And you have 3 of those that will be in the entrance of both of the major arteries. And here's just an overhead view. So here we've got a cartoon diagram of a cross section through the top of the heart right here. And so you can see the bicuspid valve here, the tricuspid valve here, and then the 2 sets of semilunar valves in the major arteries. Here you can see a semilunar valve that is somewhat open, and here you can see a semilunar valve that's almost closed, and you get a good idea as to why that makes a really good seal. And you know, if you've got if any of you have ever had a heart murmurs are most often caused by leaky valves. So the murmurs, you hear blood kind of sloshing back in a direction that's not supposed to go. All right, now let's talk about some fun stuff. The heart is an amazing organ because it is capable of functioning without any connection whatsoever to the nervous system. If you take a heart out of the body, it will continue to contract without any connection to the body, basically until it runs out of fuel. Okay. So how does it do this? And I'll tell you, I'll tell you a story in a moment to illustrate that. So atrial and ventricular contractions, they occur automatically, and then they will just be transmitted by a series of tissues throughout the heart. The contraction relaxation cycle of a heart, contraction is referred to as systole, and relaxation is referred to as diastole, and this is intrinsic, as I said. It is just built into the heart. It continuously, for the entire life of the heart, will go through this alternating state of systole and diastole. The contraction of the heart is initiated by a special group of cells that are located in the upper portion of the right atrium. That's called the sinoatrial node, or SA node for short. And these SA node cells are really amazing because they constantly are in a state. They are never in a resting state. They are constantly sending contraction signals to the heart. And we'll talk about the specifics of that. We may even get to that today. The SA node is considered to be the pacemaker of the heart because it dictates how fast the heart is going to beat. It will initiate the signals that will generate both atrial and ventricular contractions. So once the SA node sends a signal, it will pretty much instantaneously spread through the atria. The atrial tissues are thin, so it doesn't really take very long for that signal to spread rather rapidly. And then once it's made its way through all of the atrial tissue, it will be transmitted from the atria to the ventricles via another group of cells that are along the lower part of the septum of the right atrium that we call the atrioventricular or AV node. Now, some interesting things happen at the AV node. Once the signal is intercepted by the AV node slows the signal down. The idea here is that you don't want the atria and the ventricles to contract simultaneously. And this allows time for the ventricles to fill. So the rate of transmission of the signal through the AV node is going to slow down substantially. And I'll give you some numbers for that, probably not today, but next time. So once the signal makes its way through the AV node, it will then be transmitted down atrioventricular bundles, or an older name is the bundle of His. In fact, I still think the human phys book still uses bundle of His, if I'm not mistaken. And there are these big, almost like wire-like structures that are going to basically transmit the signal to the thick ventricular walls. And then coming off of these AV bundles are going to be smaller fibers that we call Purkinje fibers. And so basically the ventricular walls are totally wired to increase the rate at which the signal is transmitted through the ventricular walls. If the signal just passively isn't the right word. If the signal just made its way through the tissue itself, it would take far too long for the ventricles to contract simultaneously. And so what these AV bundles, bundle branches, and Purkinje fibers do is it creates an avenue that will allow the depolarization signal to make it very rapidly to all of the tissues in the ventricles. Does that make sense? Questions at this point?
How does the AV node slow down the signal?
Um,
Is it like a bottleneck? Effect kind of?
I'm sorry?
Is it kind of like a bottleneck?
I don't know if it's kind of like it's a characteristic of the tissue that slows down the rate at which depolarization is going to occur. The exact mechanism for that, I don't know. But frankly, all of these fibers have different rates of conduction. And I'll actually make mention of that in a second. I already said that. Got ahead of myself, I guess. All right, so let's talk about the SA node because what's going on here is just really, really fascinating. So as we've mentioned, the electrical signal for the heart begins in the SA node. The SA node is never in a resting state. So you would never measure a resting potential in an SA node cell. As soon as the SA node reaches its maximal point of depolarization, or repolarization, I should say, it will immediately begin to depolarize. And that's because of a specialized set of channels that the cells have in their membranes that we'll talk about in a moment. Now, the action potentials in SA node cells look very different than what we described in nerve cells or muscle cells. The initial depolarization that we see, so if this is a measurement of an action potential in SA node cells, initially sodium plays a role in the depolarization of the membrane. But the action potential itself is actually a result of the rapid influx of calcium and not sodium. So they have voltage-gated calcium channels, which may make sense because they really are a modified muscle cell. So what happens here is the cell will depolarize initially primarily from sodium, and then it will hit its threshold, and then you'll just get this massive influx of calcium, and an action potential will occur. Potassium plays a similar role as it does in nerve cells here to repolarize the cells, and then it just starts all over again. Okay. So the change in membrane permeability to calcium that produces the action potential, the changing potential that occurs as a result of the flux of calcium, we refer to as a pacemaker potential, another potential name. Got a few of those. Now, this is where the SA node cells get really, really interesting. The SA node cells, the reason that they never reach a resting state is because the membranes are really leaky to sodium. And so they have these interesting channels. There are these I sub F channels that produce what they refer to as a funny current. And I suppose they use the name funny current because it's just the oddest sort of thing, you know, in terms of what's going on here. The SA node cells never repolarize beyond about minus 60 millivolts. That is roughly their low point. And so that would be right down here. And when the membrane potential gets to be around that minus 60, minus 70 level, these funny channels open. So they are electrically gated to some degree. And then when you start to approach minus 40 millivolts, then we start to see the calcium channels that are going to open up. And they will open up en masse once you get past the threshold, which is about minus 40 millivolts. And this pattern, like I say, goes on for the entire life of the heart. As long as you are alive, every time your heart beats, your SA node cells are going through this entire cycle. Now, funny current involves a mixed current of sodium potassium that activates once you hit that around minus 60 millivolts. And they stay activated until you get close to threshold. Now, this is what makes these channels really interesting, is that not only do they respond to changes in voltage, but they also appear to respond to binding with cyclic AMP. So these cyclic nucleotides, and specifically cyclic AMP. And just as a preview here, it is this binding to CAMP that is used by both the parasympathetic and sympathetic nervous system to regulate heart rate. So CAMP binds directly to the I sub F channels. And what it does is it increases the likelihood that the channel is going to be open. Now, these channels are voltage gated. They probably are not all exactly opening at the same time, but if you bind to CAMP, this will get them all to open. So if they're all binding to CAMP, they're just going to all open, and they're going to all open fast. So you get from minus 60 to minus 40 threshold much more quickly if you have a lot of CAMP involved. Does that make sense? Kind of see why they call this a funny current? I mean, we've not seen anything like this before. Sympathetic stimulation will increase CAMP availability. Remember how it works, okay? It's activating those GPO proteins, which is activating adenylyl cyclase, which is pumping out CAMP. Okay. And that will increase your heart rate because it's going to reduce the time between SA node contractions, or stimulations, I should say. It's also interesting that CAMP also will improve the rapidity with which these calcium channels open. So you can really upregulate heart rate just by kicking out CAMP. And I think we'll go ahead and finish with this today. Here is a graph from one of the texts. I'm not sure which one it is. But the green line here represents a standard SA node pacemaker potential, changes in the pacemaker potential here. The yellow line here represents sympathetic stimulation, the purple line, parasympathetic stimulation. And note that the slope of this line becomes shallower as we move left to right here. So the steeper this slope here, the faster the heart rate. The shallower the slope, the slower the heart rate. Okay. So while the SA node does not need the nervous system to contract, the nervous system certainly regulates how fast it contracts. I think I'll save my story for next time. We'll start with that on Friday because it's a good story. Any questions? Isn't that interesting? All righty. Everybody have a fine day.